think we can start our session. Do you see me and do you hear me well? Okay. I'm going to start sharing my screen now. So do you see my screen? Yes?
Yes, it's there.
Okay.
Yes.
Perfect.
Good. So good evening, everyone. And thank you for joining us for the workshop Advances in Computational Modeling of Cellular Processes and High Performance Computing. So I'm Dr. Anna Nierekis. I'm an associate professor at the University of Paris-Saclay, and with Dr. Arnau Montago and Dr. Miguel Ponce de Leon from Barcelona Supercomputing Center, well, we have done our best to put together an interesting schedule for today's evening. So we have 7 excellent speakers, Dr. Rosa Maria Badia from Barcelona Supercomputing Center, Dr. Maria Rodriguez Martínez from the Research Laboratory of IBM Zurich, Dr. Jonathan Karr from the Icahn School of Medicine at Mount Sinai, Dr. Denis Thieffry from École Normale Supérieure and PSL University, Dr. Marta Cascante from the University of Barcelona, and Dr. Joerg Stelling from ETH Zurich will be giving 6 keynote presentations, and Dr. Paul Macklin from Indiana University in the US, who will be giving us a highlight talk for multiscale modeling of SARS-CoV-2. So some useful information. We are going to distribute some handouts, brief presentation of organizers and speakers with contact details, and the slides of today's presentation. You will find the link pasted at the discussion forum at the ECCB platform. Keynote presentations will last 17 minutes, plus 3 minutes for questions and answers. The highlight talk will last 30 minutes, and we will have 10 minutes for questions and answers. Questions will be addressed to the speakers only by the organizers, and please use only the live Q&A for your questions. So attendees have 2 ways of communicating, first with presenters and organizers through the live Q&A, and among you using the discussion forum. Kind reminder, presentations are streamed via the Zoom application, while the Q&A and discussion forum is on the ECCB 2020 platform, and all ECCB 2020 sessions will be recorded and stored by ECCB 2020 organizers. Those videos will be published after the Congress and will be available for the registered attendees. So thank you all for participating. Enjoy the workshop. And I will invite Arnau, who will be chairing the first session, to introduce the first speaker for today. Thank you very much.
Good. Thank you, Anna. Thank you for the nice introduction. I'm going to share screen. Yes. So our first session today will be kick-started by Rosa Maria Badia, who's a group manager at the Barcelona Supercomputing Center. So her research focuses in parallel programming models for distributed computing and also in workflows environment for different scientific applications. So she will talk today about one of those works that she has been doing with the DISLib tool that she will be presenting today. So Rosa, the floor is yours. Let me just.
Thank you very much. I will share my screen also. So, can you see my slides? Yes, I understand that.
Yes.
Thank you. So, well, thank you, Arnau. Thank you, all the organizers, Miguel and Anna, for inviting me to give this talk. Okay. Although I put this title to this talk, DISLib, Parallels in Machine Learning and its Applications to Scientific Applications, the talk covers a bit more the work we are doing in general on development of parallel applications of workflows, okay, and also how we are supporting scientific communities on development of workflows, okay. So sorry, because maybe the title is a bit misleading, okay? Okay, so a bit the outline of the talk is it's shown in this slide. So I will first introduce a bit what is PyComs, our environment for the development of parallel applications, workflows. Then I will talk a bit about the BioBB workflows, since I think it would be a bit more closer to what the community attending this workshop is doing in their research. Then I will speak about the DSLIP, and what is the DSLIP, and a bit about our recent development. I will show 2 examples of different methods that we are developing, and then I will conclude the talk. So a bit what is PyComs, what is the ambition we have? No? So what are the type of infrastructures that we've been working with? Initially, we've been working with large infrastructures like HPC, large supercomputers, and looking into exascale computing, and also with the cloud. But recently also we are seeing that the trend is to take into account what is sometimes called as the computing continuum, okay? That have devices in the edge, can be sensors that generate a lot of data, instruments. You can have edge devices like Raspberries, mobiles, a lot of small devices that can be also involved in the infrastructure. Okay, so we have that every time this infrastructure is more complex with a lot of components of different nature, different characteristics. Okay, making more difficult to deploy applications, to execute applications. No? And on the other side, also, if you speak to application providers, every time the applications are more complex, no? Because every time the scientists and the developers of applications have more tools to develop their applications, they can think of more complex applications that solve more complex problems, composed of multiple components, pieces of software that typically are described, are taught as workflows. No? So a bit our goal is how to describe these workflows, no? Tackling scientific applications and in general applications in such environment, in such complex environments. No? And what we are trying to do is develop holistic approaches where both the data and computing are integrated in a single flow, no? A single application environment, okay? With using simple and high-level interfaces. No? And also integrating workloads that include both HPC or computational workloads with machine learning and data analytics. And this powered by an intelligent runtime, non-intelligent system back in the background that can make the decisions on scheduling, allocation, data transfer, and others. No? So a bit this is what we are aiming to do. And for this, we've been working for a while with this COMS environment, okay? And well, now we have the PyComs, which is the Python binding, okay? So what is the objective of this environment? The idea is to give a simple interface for programming applications or workflows, following a sequential programming approach, because programming in sequential is easier than thinking in parallel, no? That developing parallel applications. And we aim to use general purpose programming languages that people use, like Python, that a lot of scientific communities are using. And adding some annotations, for example, in Python in the form of decorators, that help our system to do this parallelization, this execution in the complex infrastructure. So you can see an example of annotation here. This add task is our main annotation that identifies, in this case, this method as a task. No? And what is a task? Well, the task is the unit for parallelism. No? If you have a lot of tasks, then if those tasks don't have data dependencies between them, you can run them in parallel. No? And if you have 2 or more tasks that have data dependencies, no, that the result of one task is used by another task, you have a data dependence there, and you cannot run them at the same time. No, you need to run first a task that is generating this value, and then the other one, for example. No? So basically, what we do is this: we have these simple annotations, and at execution time, depending on the tasks that are involved in the code, we build a task graph or a workflow, okay, that describe the potential parallelism of the application, no, and the data dependencies that you have in your application. Okay, So with this, we can exploit the potential parallelism of the application. No? And we do this then running the different tasks on different computing resources, okay? And taking care of the different data transfers when they are required, because we run this in a distributed computing platform. No? So maybe you are running one task in one node and another task in another node, so you need to transfer data. It also offers to the programmer this view of a shared memory. So the user does not have to take care of moving the data, both memory data or files. Okay, we take care of this. And then also this has the advantage that the application can address larger data problems, no? Because if you were only able to use the memory of one node, you will be limited to that node amount of memory. But since we can use the memory of all the nodes for the application, we can address larger problems. We will see this with this lib, for example. And we tried that the code for the user remains agnostic of the computing platform. No? That the same code can be running with a smaller data set in your laptop, so you can do your tests and whatever, but can run for larger data sets in a cloud or in HPC, or when we have these other type of environments with H2 cloud systems also can run in this more distributed environment. No? And also we use container engines, Docker and Singularity mainly, to be able to manage these applications. And this again can be quite transparent to the user. Okay, the programming model is what the user will see, and but underneath there is this runtime. I will not go into all the details, but just that you are aware that basically this runtime is responsible for making all the decisions. Is deployed as a master worker application. So you have in one node the master that will run the main program and will generate the task graph and so on. And the other nodes will be responsible for executing the task. No? Once we start the execution in the master node, we will start generating tasks, and these tasks are then submitted to the worker nodes. Okay? And as you can see, well, the runtime is developed as a component system, which every component takes care of these parent aspects. Okay, and we can have both objects in memory and objects in file, and the system will take care of this data transparently to the user. Okay. Some features, no, because there are a lot of these environments, but some, I think we have some features that are not available in other systems. For example, you can give constraints to your task. No? Imagine that you have a task that requires a lot of memory. No? This happens in some of our life science applications when a task is calling a given binary, and this binary is using maybe all of the memory of a node. Even if it's a sequential task, it's using all the memory of a node. So you can use this constraint to specify that you don't want other tasks to run at the same time, so you have the exclusivity of the node for that binary. We can link with other programming models. No, we can work together with traditional parallel programming models. For example, with MPI, if you are using GROMACS is parallelized with MPI. So in our workflow, we can have sequential tasks, but we can also have parallel tasks using more than one node. Okay, in this case, you indicate the number of nodes here, 16 for this MPI simulation. Okay. And COMS is able to allocate these 16 nodes and execute them and treat this execution as a single task inside a workflow that composes more tasks. No? And recently, we have also added, for example, some task failure management, also thanks to feedback to some of our users. For example, if you are running for 200 molecules, a given simulation, maybe you don't care if some of the simulations fail, no? But you want the whole workflow to finish. No? So you can define policies on what happens if a task fails. No? Just ignore that this task has failed, or in this case, we see cancel successors. If this task has other tasks that go after them, you can say, okay, cancel this task and all the others that depend on this task. No? So because if not, at the end, they will fail. No? So these are type of things that you can indicate in the notation and help the user. No? And in general, the environment has a monitor. You can generate trace files for detailed performance analysis, and we have integration with Jupyter notebooks. Okay? So then in the framework of the BioExcel Center of Excellence, we have worked together with our colleagues in the Life Science Department and also with IRB in the development of this BioBB. Also, mainly they are the developers of BioBB, and we provide them with comms infrastructure below to be able to run large executions in HPC, for example. No? So the BioBB are some Python building blocks that enable to create a layer of interoperability to develop biomolecular simulation applications. No? So the goal is to enable the easy development of workflows, in this case already at a higher level of abstraction, no? More focusing on the problem. But internally, they generate comms tasks that are managed by the comms runtime. Okay? Some of the chief features, for example, that the BioBB is using right now is the execution of multi-node tasks. For example, they use simulations, GROMAC simulations that use more than one node, or the test failure management that I was describing before, or also task timeouts. No? You can also give timeouts to tasks if they are lasting a lot of time. Maybe you want to cancel these tasks because you know that it's not going to get to any meaningful result. No? As an example of the research that they are using these BioBBs, for example, in the framework of the COVID-19 issue, they have been working on this goal of understanding the evolutionary path that make the virus go from the bat to the humans, and to predict different aspects like the sensitivity of different people to the infection, or the impact of the virus mutations, or to predict possible new variants of the virus. No? They have been doing this with BioBB workflows, two different BioBB workflows that they have been running multiple times, no, a lot of times. So one of them is generating some simulation data, and the other one is then doing the evaluation of the impact of mutations in the binding affinity. No? And this has been resulted in a very big data study that has been run using Praise computational power. No? So we've been working together on this problem. Okay, so then on the DSLib, no? The DSLib is a parallel machine learning library that we started to develop with the goal of providing a library which solve some machine learning algorithms, but internally is parallelized. So now we go a bit higher in the level of abstraction. No? With PyComs, the user needs to think a bit of how to split the problem, how to organize the code in tasks, and so on. But with DSLib, you don't need to do this because we are offering a simple interface inspiring scikit-learn. No? Some of you, maybe you are familiar with scikit-learn that follows the fit-predict interface. And internally, the different methods are parallelized with PyComs. Okay, so and then we are not only offering this parallelism, also we have this data structure, the DSRI, which is a distributed data structure that enable us to use the memory of multiple nodes that are running a single problem in the computation. No? We will see an example where this makes sense. No? And so in this case, the parallelism is already transparent. You write your code without thinking about the parallelism. This is open source, available to the community, and provides methods for initialization of data, for example. We have multiple methods for clustering, classification, model selection. This has been done in collaboration with Fujitsu, that has been funding us on this project. Okay. So how well performs the DSLib? No? Well, one objective is to be competitive with other approaches. For example, here we are comparing with Dask ML that also follows a similar task-based approach, and also with MLlib. You can see some comparisons in the case for the case means clustering, okay, using up to 32 nodes of Mare Nostrum, that is this 1,500 node cores. Okay, so we are trying to run in relatively large infrastructure, okay, and with varying the size of the problem, no? With 1 million, 2 million samples, 500 million samples. No? What we can see here is that, DSLib is the blue line, so less, and this is time, so less is better. So you can see that, most of the cases we are better, no? Or we are better, in this case, we are better in all the cases. But not only this, is you can see here, you have some disruptions in the lines. No? Here we don't see the green line, and here we don't see any green or orange line. What this means, that for this size, DaskML or MLlib were not able to solve this problem. So in this sense, what is our added value is not only the performance, it's also that you can solve larger problems that with other systems you could not solve. Okay, okay, then a bit about our last two examples. I will go very quick. We've been working on the PCA. PCA is used a lot of times in many different problems. Okay, basically what it does is reduce the dimensionality of data sets. So we started with a parallel implementation of PCA, but that once we were getting internally, it computes a covariance matrix, and we are solving this covariance matrix with a sequential NumPy primitive. No? So this was solved sequentially at the end. What is the limitation? First, that is sequential, so it takes more time, but also that this covariance matrix, when you try to solve it and the eigen decomposition, we were running out of memory of the single node where this memory with this matrix was stored. No? So right now what we have done, we have parallelized this PCA, following a little bit different approach. Now we are using an SVD to find the values, the eigenvalues, okay? But now it's running in parallel. For example, in this case, it's running with 6 Mare Nostrum nodes, and we were able to solve for a trajectory with 6,000 snapshots of almost 7,000 atoms. And this problem we were not able to run with this previous implementation. No? So these are the type of things that we are trying to do now in collaboration with our life science department and IRB, no? To see which are the problems that are interesting for them and that we can help. No? Similarly, I will skip the description, but we are working on the Gromos clustering. We have an early implementation. Still, we cannot say if it's competitive or not with a traditional implementation, but the advantage will be that it's parallelized and that can run on multiple nodes of an infrastructure. Okay. So a bit to conclude my talk, we try to offer this general purpose task-based programming model. However, it's not only the objective to provide this environment, also is to help other groups like the life science to build these other BioBB or other systems that can run on top of PyComs, okay, and be offering them to exploit larger systems, or also working in things like, for example, the DISLib, which is more a product, a final product that scientific groups can use, but that at the same time they will allow to run in parallel their applications. Okay. Okay, you can find more information in our project page. Everything is open source. We have Docker images, we have example applications, and so on. For any question, you can. I didn't put here the email address, but I can send it to you later. We have a global group email address which many people answer. Okay. Thank you very much. I spoke a bit longer. Sorry.
Okay, thank you, Rosa. Virtual applause for Rosa. Thank you very much for the talk. Very interesting and very clarifying. I have was questions from the audience. Oh yes, if talking about the DISLib tool that you presented, could it be somehow similar to running scikit-learn on something like TensorFlow? Or what is, I mean, can you trace a parallelism there?
Yeah, with TensorFlow, the approach we are following is to work together. I mean, it's not that we substitute TensorFlow. The idea is that we are ongoing work to integrate TensorFlow with the DISLib, no? In the sense that we can use this DS array, the parallel and distributed array, to have the data distributed. We are working with TensorFlow and other approaches. There is a group at UPP in Valencia working on a parallel version for solving also neural net or so on. And what we are trying to do is, for example, approaches that can do asynchronous evaluation. So we reduce the synchronous steps that we have after every epoch, okay, enabling then the system to go a bit faster when evaluating different epochs. Okay. Okay. So we have been working, for example, with our AI group at ESC, integrating TensorFlow in one application that was called Tiramisu. In this case, TensorFlow was launched as a task, for example, and then we were doing some other analytics with PyCons. Okay. But this was not with the sleep, but it's the kind of things we can do with integration of TensorFlow.
And talking about the data deconvolution the work on the data deconvolution, the PCA that you presented at the very end, are you planning to extend it to other data deconvolution that are also sequential, or that at some point?
Yes, but since this is very interesting, because if anybody is interested in any specific algorithm or in any specific method, please let us know, because we don't have infinite resources. Okay, so we try to be effective in this sense. No? In this case, this PCA, we speak to some of our colleagues and they say, yeah, PCA can be interested. So we did the first implementation. We saw that we had some problems, and then we are doing this more parallel implementation. So if anybody has suggestions of things that would be useful for them, please let us know, because then we can put it on the priority list of things to be developed. Okay, yes, we try to increasingly be adding different methods, different algorithms.
Yes, and then just a curiosity, in the BioBB project, the implementation of comms that you use, is it PyComms or is it the?
Sí. The BioBBs are in Python. Okay. Sí. In fact, they can run independently of PyComms. Okay. They can run the BioBB sequentials, and they can run with other workflow environments. And they run with us, especially when they use supercomputers, okay, to enable more parallelism. Mm-hmm.
Okay, good. Thank you very much for your presentation, and we'll move now to Jonathan Karr, who will be talking about whole cell models and novel technologies in order to model whole cells. So let me just share the PDF. Yes. So Jonathan Karr is an assistant professor at Mount Sinai in the Institute of Data Science and Genomic Technologies. So his research focuses, as I said, the whole cell dynamical modeling, and also computational methods for large-scale data-intensive modeling. So I think I posted the link to this presentation and all the presenters' presentation in the discussion forum. So if any of the attendees are interested, they can go there and download this presentation. So, Jonathan, the floor is yours.
Great. Great. First, I want to thank the organizers for inviting me and for continuing to organize this online, even during the pandemic, and making it accessible to people around the world. So I'd like to talk about some technologies that we've been developing to make it easier to construct more comprehensive models of individual cells. So from our perspective, we believe that more comprehensive models of individual cells will likely be a key ingredient to the advancements of precision medicine and synthetic biology. As we've learned over the past decade, we know that each individual has a unique genome with genetic variations scattered throughout their genome, and in principle, this can occur in any pathway inside a cell. So we know that furthermore, this impacts how disease progresses in individuals. We know that this impacts how individuals respond to drugs. And so we believe that we need more comprehensive models that help us reason about all this genetic variation, which is scattered throughout biochemistry, to help physicians choose the best therapy for each individual. Similarly, we think that we will need more comprehensive models to help us use computer-aided design tools to design microorganisms, cells, tissues, higher order systems for a broad range of applications. So we think that we need to build models of entire genomes. We think that we need to build models that represent all of their biochemistry. We think this has to account for their entire life cycle. We also think that we need to build mechanistic models that allow us to utilize universal physical principles to extrapolate from a few well-characterized organisms to all possible organisms that we might want to design, or all possible patients. So several years ago, we began to explore how we could build more comprehensive models of individual cells, focusing on Mycoplasma genitalium, one of the smallest known freely living organisms. And what we've been able to show is that, at least for a very small organism, we can build a model that at least coarsely captures the annotated function of each gene product. So we achieved this model first by integrating a broad range of data about Mycoplasma genitalium and other gram-positive bacteria. We then use this data to construct submodels of each subsystem inside Mycoplasma genitalium. We use different mathematical formalisms to capture these processes at the scale appropriate to each process or subsystem. We then combine these submodels into a single multi-algorithm model, and then developed algorithms for co-simulating these mathematically heterogeneous submodels. Now, despite this progress, there's still a variety of biochemistry that our model doesn't capture. There's a variety of phenotypes that our model cannot accurately predict. So over the past few years, we've been challenging ourselves to try to develop more comprehensive and more predictive models of small bacteria, focusing on Mycoplasma pneumonia. And we've tried to challenge ourselves to build equally comprehensive models of much more complex organisms, focusing on human embryonic stem cells. So in order to enable more comprehensive and more predictive models of cells, we've been reflecting on this workflow and trying to figure out how to scale it up to more comprehensive and more complex models. In addition to reflecting on our own experience, we've also, a couple of years ago, conducted a survey of the biomodeling community to get input on the community on where we should focus our attention to enabling more comprehensive and more predictive models. And this has led us to focus on a few specific bottlenecks which we believe are the biggest barriers to progress. First of all, the most frequently cited barrier from the community, and which we believe is also a key barrier to progress in wholesome modeling, is lack of access to the data that's needed to build or calibrate or validate models. Second, at the moment, while there's a variety of frameworks or formats for systematically and clearly describing biochemical models, at the moment, it is difficult to systematically describe hybrid or multi-algorithm models that utilize multiple modeling frameworks simultaneously. This makes it difficult for modelers to have detailed conversations about hybrid models. This also makes it difficult, say, for peer reviewers to review in detail a model closely related to this, because there's no systematic representations of, or minimal systematic representations of hybrid models. It is also difficult to systematically simulate hybrid models. We also believe that underlying all this, it remains still too difficult for modelers to work together to build models or to simulate or to apply models. So over the past 2 years, we've been trying to develop practical solutions to these major bottlenecks to progress in wholesome modeling. So over the next few minutes, I want to tell you about some of the things that we've been building to make wholesome modeling more possible. So let's first consider an example. Let's consider trying to build a dynamical model of metabolism. If we want to model metabolism, we would likely need at a minimum some data about concentrations of metabolites, data about abundances of enzymes, data about the rate constants of reactions. And if we want to model an entire cell, we need vastly more data than this. We need likely data about sequences of chromosomes, locations of promoters, locations of terminators. Now, we have a huge amount of data available about molecular biology. That said, I think it remains difficult to gather the data that you need for something like a wholesome model, because our data is scattered in many different databases, in many different articles. It's described with different formats, with different identifiers, with different units. And so it's difficult to compose heterogeneous data into some sort of integrated data set, which could be used for for modeling. So to address some of these issues, we have developed DataNator, which is available at datanator.org, a toolkit that helps modelers or other investigators find data about specific biochemical mechanisms, so specific species or particular reactions in specific organisms and specific environmental conditions. The first part of DataNator is an integrated database of several types of molecular data. Initially, we focused on what we feel are the most essential types of data for wholesome modeling. Currently, we have data about metabolite concentrations, data about modifications and half-lives of RNA, data about modifications of proteins, data about rate constants of reactions. We've already integrated data about RNA half-lives, RNA localizations, protein half-lives. That data is integrated in the database and will be coming soon to the web interface for DataNator. We're currently working on curating data about reaction fluxes and hope to get that out to the user interface soon as well. Now, on top of this integrated database, we've developed workflow or software tools for helping modelers discover data for specific modeling projects. Like other databases, this begins by users conducting a full text search where they search for data about a specific metabolite, RNA, protein, or reaction. And then users are presented with a list of species and reactions for which we have data. So once a user then chooses a species or reaction that we have data about, they then are shown tables of measured properties of that metabolite, RNA, protein, or reaction. Now, one of the challenges, I think, to finding molecular data is that we don't have complete data for every metabolite, every RNA, every protein, every reaction, in every organism, and in every environmental condition. However, we do often have data for chemically similar metabolites in taxonomically similar organisms in similar temperature or maybe similar pH. But for the most part, it's difficult to search for such data using, say, Google Scholar or even more biologically oriented databases like OmicsDI. So DataNator tries to approach this problem through a couple of tools. First, DataNator builds broad clouds of data which encompass potentially relevant data about the species or reaction that a user may be interested in. It does this by including data from chemically related species and reactions and from similar environments and organisms. And then DataNator provides users a set of filters which they can use to narrow down these broad data clouds to just the set of data which a user believes is most relevant to their research. So for example, a user can narrow down a broad cloud of data to only data to only observations of highly chemically similar metabolites as scored by their Tanimoto coefficient. Or if a user only wants data from E. coli and closely related organisms, a user can use a slider to show only data from the same phylum or the same family or the if you want to go as broad as possible, the same kingdom. Now, so far, we've integrated data into DataNator by manually developing codes to pull data from different sources, normalize it, and integrate it. One of the long-term challenges to doing this is to get data generators to provide more structured data, which can more easily be incorporated into something like DataNator. So to address this issue, we've developed object tables. This is available at objecttables.org. It's a set of tools which enable authors to create higher quality, more reusable spreadsheets, which can be published as supplementary materials associated with articles. Basically, object tables makes spreadsheets or enables authors to create higher quality, more reusable spreadsheets by combining spreadsheets with schemas. And then on top of that, object tables provides authors software tools that they can use to use schemas to error check spreadsheets. In addition, object tables also provides like an object relational mapping system, which enables users to take spreadsheets and map them into object-oriented data structures, which then can be easily manipulated with Python or other programming tools and easily incorporated into a structured database like Datator. So we think that object tables is a very powerful and fairly simple set of tools which can enable authors to create higher quality, more reusable supplementary materials across a broad range of science. So as I mentioned previously, we think that likely one of the most effective ways to build wholesome models will be to combine models of different cellular subsystems which are described using different mathematics. In addition, we believe that wholesome models will have to capture the combinatorial complexity of biochemistry. So they'll have to be able to capture all the transcript variants of each gene. They'll have to be able to capture all of the phosphorylated forms of each protein and so on. So basically, this means that we need to be able to systematically and clearly describe composite algorithmic models which are described using the very abstract reaction rules. So to enable this, we've developed WC-Lang. This is available from our GitHub organization, a new language for describing such composite hybrid models. And a key piece of this is that it enables us to describe models using more abstract rules. Just to give you some intuition, you can roughly think of this as like another layer of abstraction on top of rule-based modeling systems like BioNetGen or Kappa, which are already abstractions on top of reaction network formats like SBML. And basically, this allows us to compactly describe very complex reaction networks using comparatively simple reaction rules. These rules then in turn enable much more modular descriptions of different subsystems of biochemistry, which in turn facilitates collaboration. So in order to interpret models that are described using WC-Lang, we've developed WC-Sim, a tool for co-simulating models or submodels which are described using WC-Lang. Now, one of the reasons why we think it remains difficult for modelers to collaborate on models is that it's difficult to precisely describe the semantic meaning of each species and each reaction represented by a model. This is because typically models are annotated relatively coarsely, either using IUPAC or FASTA sequences or UniProt identifiers. But the problem with IUPAC and FASTA and UniProt identifiers is it doesn't have sufficient resolution to be able to describe individual phosphorylated forms of proteins, for example. So to address this issue, to allow us to more precisely describe the semantic meaning of models and in turn facilitate collaboration, we've developed BP forms and BC forms. You can roughly think of BP forms as like a generalization of IUPAC or FASTA, which allows us to describe the entire primary chemical structure of DNAs, RNAs, and proteins, including hundreds or thousands of non-canonical nucleic and amino acids, crosslinks, nicks, circularity in DNA, everything that encompasses the primary chemical structure of macromolecules. Now, to further facilitate collaboration, we're also developing a platform that we're calling biosimulations, which will enable modelers to share not only modeling models like biomodels or big, but entire modeling studies, including simulations, data visualizations of simulation results. And as part of biosimulations, we want to make it possible for modelers to be able to reuse models that may be published by other people. One reason why we think that this is currently difficult is that there's a sea of modeling frameworks. There's many different simulation algorithms. There are a variety of different modeling formats. This is, I think, difficult, especially for non-experts to navigate. So as we want to try to involve experimental scientists more directly in modeling, we need to provide tools that help people navigate this complexity. So to make it possible to make it easier to reuse models, we are also developing a registry of containerized simulation tools, which all provide a consistent interface. This is available at biosimulators.org. We currently have several standardized simulation tools that encompass some of the most popular simulation tools for biochemical modeling. Going forward, we're going to be using these containers to provide a simple, consistent way of accessing a broad range of simulation methodology through a simple web-based user interface integrated into biosimulations. So just to quickly summarize, we think that this combination of technologies will lower some of the biggest barriers to wholesome modeling and in turn enable more comprehensive and more predictive models. Going forward, we hope that this will enable models which help physicians make decisions for medicine, as well as ultimately help synthetic biologists design a broad range of biological systems. So I just want to thank the members of my group, and with that, thank you very much for your time and attention.
Thank you, Jonathan, for your talk. So you have virtual applause from here. I find it very dense, and I mean, you presented several, I mean, many different tools and different aspects of simulation. I go on. So waiting for people to use the Q&A, the live Q&A. I have here questions. So there's one question from the one attendee. Given the fact that the nanopore provide access to the raw signal at molecule level, do you think BP forms could be compatible to incorporate nanopore signal data to perform the simulations for molecules modifications?
So, does this refer to modifications of DNA? I
guess so. So molecules modification, if it's nanopore, it should be, yes. Yes.
Yeah, so this does allow us to describe, so if the IOPAC nomenclature, what most people think of as FASTA, allows us to describe DNA that's composed of the 4 canonical nucleic acids. But in reality, DNA, RNA, and protein are composed of many more nucleic acids beyond just the 4 canonical ones. So this allows us to capture that biochemistry. There's a lot of different ways which it can be measured. For DNA, one possibility is deep sequencing based methods. For RNA and proteins, there are other methods that are typically used to conduct this information. So this basically provides a way of integrating these different sources of information, using this as a way to annotate the semantic meaning of models, and in turn, making it easier to more precisely model biochemistry.
Okay. I had also one question about your object table. Do you link it somehow to further data or to the, let's say, the identifiers that further data wants people or encourage people to use?
Yeah, so object tables supports a broad range of data types. There is a data type specifically for ontology terms, and then this can be linked to any ontology that's in, say, BioPortal or OntoB or anything like that. So you can embed any identifier that you want. There's also a data type for identifiers. If you want to link to like identifiers.org, you can do that, and then the identifiers can be validated.
Okay. There is also another question about the datonator. So imagine that you want to create a database of favorite cell line or microorganism because you're interested into modeling this cell line. And could the tool be used to organize and fetch molecular data and parameters for this given cell line? I mean, that is not already included. And if yes, how hard would it be to, let's say, expand datonator to other organisms or other cell line?
Yeah, that's a great question. So our philosophy has been that we actually don't really want to create a database of any single organism or any one specific cell line, because we think that at least from our perspective, the data that we need is typically too sparse for this to really be useful for our purposes. So usually, I think I disappeared there.
Yes,
usually for usually for our purposes, we need data from multiple cell lines or even often data from multiple organisms. So we think that it's useful to just compile all the data into one place and then provide users tools for filtering out data for one specific cell line. So then instead of having a proliferation of databases for different cell lines, you just have one database and then filters, which will allow you to effectively, if you want to think of it, create like a virtualized database of an individual cell line from a superset that encompasses all possible cell lines.
Good. So I think we're right on time. So thank you very much, Jonathan, and thank you very much again, Rosa, for your talks. We will now move to the second session, chaired by Anna. So Anna, I will give you the virtual mic.
Okay. Thank you very much, Anna, and thank you, Rosa and Jonathan. So for the second session, okay, we are going to start with a talk from Professor Denis Thieffry from École Normale Supérieure from the PSL University in Paris. So Denis is a professor of systems biology, head of the computational systems biology team in the Institute of Biology at the ENS. His research is mainly focused on the development of the software GeneSim for dynamical modeling of signaling and regulatory networks, and the development of the software suite ESSAT, devoted to the analysis of functional genomics. He is also very involved in applications to regulatory networks, controlling cell specification, differentiation, and reprogramming. And today he's going to give a talk focused on T-cell commitment. So Denis, thank you very much for accepting to be here, and we are ready to hear your talk.
Okay, thank you, Anna. Thank you to the organizer for inviting me. Let me share my screen, desktop, share, and then my slide. Yep. Is it working? Is it okay? Can you see my screen?
Yes.
Okay, perfect.
Yes.
So yes, I'm going to dig in more into the details of building a model. In fact, it's still an ongoing project. So we had a first publication recently, which deals with the trying to have a predictive model for T-cell commitment, so T-cell development in the thymus. It's a relatively complex process, which starts with multipotent progenitor entering in a specific organ, the thymus, and then going through several specification steps where the progenitor is progressively losing some of its potential to give rise to different cell types. And this happens, in fact, in a microenvironment providing notch signaling, so from stroma cells. And it's this signaling environment that is supposedly instructive to, let's say, force the progenitors to commit themselves toward T-cell cell fate. But as you can see in this cartoon, there are also other potential fates. Initial progenitors can give rise to other lymphocyte lineage, could be also B cells and so on. I will come back into that with the next slide, where again you see this representation where you start from hematopoietic stem cells and which get committed into a bit more already specialized progenitors before entering into the thymus, and then upon notch signaling, stepwise process with more limited developmental potential. This process has been studied in detail, in vitro, in vivo also, in particular on the mouse model. And you can find in the literature, in particular, for example, in review by the group of Helen Rothenberg, drawing or cartoons showing that some specific regulatory circuits are involved in these different steps. So you always see notch signaling as a key input, and then this signaling is interacting with or acting on specific pathways, ending up on transcriptional factor. So in green, transcriptional factor involved in T cell development or require T cell development, or in blue, transcriptional factor involved in B cell development, in orange, in yellow cell development, and so on. And so what is supposed is that these combinations of regulatory circuits is at work to progressively commit the cells in a specific cell fate. But the problem is a bit to understand how these work together. So the aim of this work was to integrate all available data on T cell development and alternative fates, in particular information about who are the main actors and how they interact with each other. And then through modeling, gain some rigorous assessment of this knowledge, and in particular identify gaps in current knowledge. And so to do so, we use dynamical modeling, and more specifically, special brand of logical modeling, multi-level logical modeling. I want to mention that this was the work, in fact, of mainly of two people who started this in my group, and then we continue in collaboration. Elisabetta Cacace, who did an internship with us and now is doing a PhD at EMBL, and Samuel Colombet, who did his PhD with me and is now doing a postdoc at EMBL. And so first, what are the main steps when we try to develop a logical model? So the first thing is to try to delineate the regulatory graph, which is very much a bottom-up thing. So from the knowledge of some specific regulatory mechanism, we draw a cartoon, and we try to get a more abstract and rigorous representation of these cartoons. The second step then is to identify maybe the key component that may need to be represented by multi-level variable. Otherwise, by default, we consider every component of a regulatory network as a simple Boolean device. And then we work on the dynamical analysis, so starting with stable state identification or other attractor identification, then the analysis of the reachability of these attractors from specific conditions, and finally simulations to try to recover more transient phenomena, and for that we use increasingly stochastic simulation approach. Now, for sure, this is usually going through some iterations. So we start with a relatively limited regulatory graph, and we go through all these steps, and we end up refining more and more the models and going through these different steps several times. I cannot do that in 50 minutes. So I show you the regulatory network that we have built using the software GenSim. And so it's not so large, but it's pretty complex in terms of regulatory circuits, feedbacks, and so on. It encompasses 56 nodes, most of them Boolean, and in fact only 5 which have been defined as multi-level nodes. You can see that they've been colored, depending on which cells they are associated, which cell fate. So the T cell program are in dark blue, the notch signaling in light blue. You can probably not read any names from where you are, but you can check on the PDF if you want. The B cell program components are in blue, and then the myeloid components are in orange, and the NK and ISC program in red. So this is not maybe so important. So that's 2 minutes only. Okay, so I will go very quickly to the results. We go through, we make use, in fact, of different tools. First, we identify the stable state, and we could identify stable state associated to these different cell fates. And then we use the software MABOS developed by colleagues. Yes, yes.
I don't know who rang the bell, but you actually have 6 minutes more. So
okay, so I was very surprised.
Yes, so take your time.
Okay, so yes, maybe I go back one slide then. So we could easily identify with efficient algorithm the stable state of the system again. And then we made some reachability analysis that I'm omitting here, just to check that this stable state can be reached from, let's say, realistic initial conditions, so corresponding to multipotent progenitors. And then I will show you some results obtained using the MABOS, so to perform stochastic simulation. For that, we have to translate the multi-level model into Boolean one, define up and down rates, but in fact, we limit ourselves to consider equivalent concurrent rates. And so we took advantage of the MABOS package developed by our colleagues, Laurence Calson at Institut Curie, and Gauthier Stoll now at INSERM unit also in Paris. And this is greatly eased by using the common logical modeling toolbox that integrate, in fact, many tools devoted to logical models. So you can check the website and the list in more detail if you want. And so you can convert model from one format to another, and that way combine different tools. So that we took advantage of that. And this is developed mainly upon the supervision of Aurélien Aldi. So just a few examples of our results, current results for stochastic simulation. So here, starting from some initial conditions where we put two inputs, two signals, one pushing towards the B cell fate and the other pushing towards the T cell fate, you see that we get some proportion of these different fates that cannot be taken directly as a real proportion, because the rates have been put in a pretty arbitrary way. And we can also follow the level of activations over time of the different components. But what is interesting is that we can compare the results, for example, for different input conditions. And you see that here, if you don't provide delta, you lose the T cell fate completely, which is in fact expected. And we can also compare what is happening in wild type situation with different perturbation. So, for example, loss of function, in that case of a key T cell developmental gene, but which is a terminal one, BCL11B, which still give rise to a T cell-like fate, but less mature one. Whereas a gain of function will increase significantly the proportion of T cell fates. You can for sure also perturb other components like KT3, loss of function, gain of function. Maybe I'll skip that one. And you can also consider situations that enable reprogramming of cells. So for example, here, the ectopic expression of 2 genes specified at the beginning of the simulation will lead to macrophage cell fate instead of lymphoid cell fate. So to conclude, the use of collomotor Docker image and notebook greatly ease the analysis of sophisticated analysis workflows that combine different tools. So in that case, I show you only 2 tools, but we use more in the complete story. In addition, the use of such notebook foster reproducibility and enhance the reusability of models, which I think is very important. So people can take this model and then try to extend it, maybe to cover or to include new data or to cover new cell fate and so on. Our current model recapitulates key aspects of T-cell specification as well as of alternative cell fates. But we have to say that at this point, it does not really enable to exclude, for example, B cell fate early enough, according to published data. And so this points to some missing regulatory components or interactions. And currently, we are working on the analysis of single-cell RNA-seq data to infer novel regulatory links that could then be tested through integration into our model. So my last slide, open challenges. So one is, as we have more and more molecular maps, is how to translate them into regulatory graph. Anna would be the good person to talk about that. Another challenge is to select the right logical rules, maybe to explore variations of these rules. And a final one, one more, is to assess model properties in a more automatic way. We did some work on that, and we could discuss it later on. And another challenge also is to connect qualitative and or go from qualitative to more quantitative results. Stochastic Boolean simulation already is a step in this direction, but we can go further for sure. So I stop here. We have one recent publication on the subject, but this is not the end of the story. Thank you for your attention. Hello.
Hello. So thank you very much, dear Denis. Virtual applause, as Anna said. Thank you very much for your talk and all these results. So we have some questions from biological observations of activated genes. Is it always corresponding to stable states in models, or is there biological observations of transient states?
There are 2 aspects to that. One is whether, for example, a cell fate need to be represented into by a stable state. Not always, because it depends very much on the model and what exactly you try to represent, but it could be a more complex attractor. And we have tools also to predict them and to analyze them, and you can also identify them through stochastic simulation. The other question maybe is sometimes you have very interesting and important transient observation, and using, for example, model checking or simulations, you could also try to recapitulate such transient event. So a very good example is cell cycle, where you know that you need a very specific succession of molecular events, and you want to capture this with a model.
The second question is, what kind of data can you use to fit the T-cell modeling, and how do you actually use the single cell data?
Yeah, so for the setting of the model, we use mainly gene expression data. So we had more bulk expression data from in vitro experiments. Plus, so that was for the dynamics to check if our model had the right dynamical behavior. But now to predict novel interactions, we use network inference tools based on single cell RNA-seq data. So there are already several approaches to do that. Now the problem is that usually you infer many interactions, and I think that mechanistic modeling can be a framework to help select between these interactions.
Okay, and a last question with a very brief answer. What criteria did you use to categorize the steady states into each of the cell fates that you showed in the presentation?
But this is well established in the literature. We know who are the key marker genes that are supposed to be expressed in each cell type or cell fate, cell progenitors.
Okay, so, Denis, thank you again. We need to move on. So I would like to, thank you very much. So I would like to, if you can stop sharing your screen, I will try to smile. Thank you. So do you see my screen now?
Not really. We see.
It's coming. It is coming. Yes.
Okay. Yeah.
Okay.
But we see a presentation mode, but okay, it will work.
You see the presenter's mode?
Yeah.
Just give me.
Anyway, we see Maria's slide, so you can go on.
So we will be welcoming Dr. Maria Rodriguez Martínez. She's a technical leader of systems biology at the IBM research in Zurich. And Maria is focusing on multi-scale models of the immune system, IAD and drug modeling and personalized medicine models, characterization of cancer heterogeneity through single cell analysis, and multi-omic approaches in systems medicine. So today she's going to give a talk about depiction. So Maria, the floor is yours.
Thank you very much, Anna. Thank you for the invitation and really appreciate the opportunity to present our work. So let me share my screen. And I guess you are seeing now the presentation. Yeah, so in this talk, I'm going to be talking about something a bit different from what the description of the topics that you have just listed. So this is an activity that we started like 2-3 years ago, and in the area of interpretability, for mostly for deep learning, but also for machine learning. So as this topic may be different, quite new for some of you, I'm gonna just tell you a little bit about what is interpretability. So this is a very simplistic cartoon, but I think it illustrates the concept very well. So this is the world that we want to model with computational biologists. And to model this world, we just usually measure some data, and then we build models. In recent years, especially since the development of deep learning, these models have become increasingly more accurate, which is really nice, but also increasingly less transparent. And this lack of transparency has usually bad implications because we are not able, first of all, to understand the underlying mechanisms. So we are failing to learn maybe useful insights, but also there will be biases hidden in the model that we are not able to capture. So because people have been realizing the problems of using models from which we don't understand the underlying mechanisms, the field of interpretability, mostly again for deep learning, but in more generic sense for the whole artificial intelligence, has been literally booming in recent years. So to say that briefly in 2 main topics, I guess I will summarize that interpretability has 2 main aims. The first one, as I mentioned, is understanding the decision process, and if possible, the understanding getting new insights from the model and from the data. But also importantly, as I mentioned, is bias identification. And this bias identification might seem a bit abstract, but this is things that have happened in real life, in real data. So as to mention an example, maybe outside of the realm of biology, but there was some of you might have heard about the model COMPASS that were used by the US criminal system. So the idea was just to predict the chances of people to commit crimes. And that was decided in order just to, in the court system. So that was really used. And what happened is as the data used to train the model was ethnically biased. There were much more Afro-American records than Caucasian. So the model basically learned that the biggest predictive factor was their ethnicity. So as you can imagine, this is a non-desired outcome of the model. So for this reason, it's really important to understand why a model is making a decision in order to understand the mechanisms, but also to pick up possible biases. So before going into telling you a little bit what we have done in this area, I would like just to give you a very brief overview of what people are doing. And as I said, this field is really booming in recent years, and people have developing their new models published almost every day. So just briefly, of course, I don't have the time to go through all of them, but I can tell you a little bit how they're roughly categorized. So first of all, modeling can be local. So by local, that means that you focus on explaining a single example. So that means suppose that you have a model that tells you whether a cancer sample or a tumor, a sample, whether this person has a cancer or has a benign tumor. So you might want to have an explanation for this particular person. So they will tell you maybe this is a benign tumor because these and these markers are not expressed. Sometimes you might want to have a global model that maybe gave you general rules that apply to all samples. So each approach has pros and cons. The local approach usually typically is easier to extract, but is less generalizable. The global approach sometimes is more generalizable, but ironically sometimes can be extremely non-complex. So to give an example, that would be an example of the first category. Here we are trying to explain what are the features of this biopsy that we think might be cancer-like. A global could be, for instance, a global model could be, for instance, a decision tree. As you can imagine, if you have many samples and features, this can be extremely complex. A different categorization, but also they're quite useful, is to keep in mind is ante-hoc and post-hoc methods. So ante-hoc typically is like models or maybe modules or parts of that you can integrate into the model pre-training, such as once the model is trained, you can look at these components and just get some insight about why the model is doing what it's doing. And post-hoc is something that you do a posteriori. And I'm going to give you now a few examples of each category, and hopefully you can see the difference. Okay, so now I'm starting to move into work that we have done quite recently. We submitted it last year to a workshop in EURIPS. And the idea we're thinking about developing ante-hoc models. So just trying to say how you can maybe modify the structure architecture of a network in order to make it more interpretable. And the basic idea was quite simple indeed. The idea is that typically we understand things that are monotonic. And by monotonic, I mean this could be, for instance, the input and this will be the output. So again, coming back to the question of cancer, so imagine that, let's assume a very simplistic cancer that only depends on the expression of a protein. So typically you could understand easily the role of this protein if the output, so if the patient is more aggressive, the higher the expression of the protein. So there's a monotonic relationship between the expression of the protein and the aggressiveness of the phenotype. So how did we implement this? So actually we were exploring the idea of a structure in changing imposing some constraints in the network in order to make it monotonically, the weights monotonic for some particular layers. So the idea is, without going into the details, is that if, for instance, you want to explain this layer, you can somehow constrain the activation functions on the posterior layers in some type of maybe complex but monotonic function, and that will allow you to give you some insight about the role of these internal features. So if you want to know the details, actually what we did, it was rather a simple approach. We just assume that the activation functions depend on exponential functions of the input of the previous layers. And this very simple kind of naive approach actually was enough to give us some insight, at least about these internal features. Okay, this is good, but actually this still is giving you only a very rough, not even explanation, it's just giving you some insight about some abstract feature in the internal layers. So an alternative approach that also is has been quite popular and used in many samples is what's called attention mechanisms. So here the idea is that, again, you will put what is called an attention layer in your network, and then you do your training and whatever you want to do with your network. And actually, once the network is trained, you can look at this layer and look at the scores. And this score will tell you which feature of the data was more informative. So for instance, this is again some work that we did. We published last year in Molecular Pharmaceutic. So we developed this model, we call it Pacman. the idea is that we want to predict how sensitive a particular cell line is a particular drug. So we use some deep learning approach and we use some attention layers. So here you have an example of the type of output that this model can tell you. So here we are predicting the sensitivity of these two FDA-approved drugs, masitinib and imatinib, on a leukemia cell line. And actually you see that we are doing relatively quite well. So the predicted IC50 value is quite close to the true value. But the most interesting thing here is that then using this attention layer, we can ask the model to tell us which patterns of the chemical compound were more informative to make the prediction. And this is quite nice because these two compounds are very similar. The only difference is in this atom here, which I don't know if you can see, but this is a sulfur. And actually you can see in this drug that is the slightly more efficacious drug, the attention layer picked that all the most informative pattern was in this particular atom. So that was a very nice validation that actually we are picking something that is informative to make the prediction. And if you would like to play with this model, actually we have implemented a web service. It's completely free, so yeah, you don't even need to log in. And you can either upload your own cell line or even your compound. It doesn't have to be a real one. It can be something that you have designed, and you can just get predictions, and you can also get this type of attention scores and attention plots and play with them. Okay, now I'm going to move to the second type of methods. As I mentioned before, you have the anti-hoc that I discussed so far, but you also have the post-hocs. What is that? Remember, so sometimes you might get a model that is already trained by somebody else, and you don't want or you cannot reverse engineer the model. So you want to have some techniques to somehow, without opening the model, to just get some insight about what this model is doing and why. So there are many approaches to this type of analysis. Here I'm gonna, of course, not gonna do an extensive review, but I'm gonna mention maybe one of the most common ones, perturbation methods. And I'm illustrating how it works with this kind of very visual example, even though it's not from the biology. So this is a model that was trained to classify images. And as you can see, it correctly classified that this is the picture of a snake. And then there were this perturbation method, this occlusion method, which idea is very simple. You just start blocking different pixels. And when you block the pixels of the snake, you will see the classification deteriorates. And then you will conclude correctly that this is the pixels that code the snake. But you start seeing the first problem here. And the problem is like you can do different types of perturbations. So here we are changing the size of the square that we are occluding. So here is a pixel, it has 1 by 1 pixel, 5 by 5, 10 by 10, 15 by 15. And you see that only in the last type of perturbation, when the square that we are blocking is larger, we correctly pick the shape of the snake. And in this particular case, actually, it's obvious. You see the snake right away, so you see that this is the correct answer. But in so many cases, actually, you don't know a priori what is the right explanation. So it might be difficult for you to assess what is a good or a bad explanation. And that tell us to the challenge that actually I would like to mention and highlight, that there's many people thinking about how to evaluate a good explanation, but indeed, as far as I know, and the community seems to agree, there is not such a thing as a universal explanation that everybody is gonna accept. This is maybe the most abstract part of my talk, so I'm not gonna go too much into the details here, but I just wanted to mention a little bit what is the problem with evaluating or ranking explanation. There's no such a thing as ranking, but just evaluating explanations. The first thing is like there is not a concept of this is the right explanation. And people have been thinking at the beginning, people maybe a bit naively, they were thinking that you can assess what is a good explanation when you see it. This type of reasoning has many problems, and the main problem is this human bias. We assume that you have, as a human, you are not biased, and you will be able to pick this is the best explanation. But as we saw before with the question of assessing the risk of committing crimes, there might be biases which are completely ignored that we have. So it's very dangerous to think that as a human, you don't have cognitive biases. So we're thinking in this paper that we just put in the archive very recently, we're thinking about how we can assess the quality of an explanation. And as I said before, the first thing is there's no way of ranking them, but at least we can define what are some good criteria that a good explanation should have. And there are a few things that seems to be intuitive that a good quality explanation should have. For instance, simplicity. So if we have an explanation that is simple and an explanation that is complex, obviously we prefer the one that is simple. it also should be broadness in the sense that it should be generalizable. We would like the explanation to cover as many samples as possible. And also we like the model to exhibit as much fidelity with respect to the predictive model as possible. So with this type of conceptual thinking, we define a few metrics of at least desired qualities that a good explanation should have. And we divided them into two different categories. First of all, the way we evaluate the part of the model that extracts the features. And for that, we define the simplicity and the broadness. I mean, we assume that the extracted features should be at least as broad to cover as many samples as possible. And then we also were thinking about how to define other qualities, characteristics of what a good explanation should have. And here I'm not going to go too much into the details. I would like just to mention intuitively what these metrics are about. So for instance, monotonicity. I mentioned a little bit in the previous example when I was talking about this monotonic network, that this is a desired quality, but here monotonicity means a slightly different thing. But monotonicity here in lay terms means that we expect that if a feature is very important, we expect that if we remove this feature, we will lose a loss of information. So actually we expect a monotonic relationship between the importance of the feature and the loss of information. So it goes in this direction. And in a similar way, we can also define non-sensitivity, the expect that the features that are not important, the model is not very sensitive to them. So this is again, like it seems kind of a reasonable qualities that we can formalize as metrics and apply to the network. So again, I'm not going too much into the details. If you want, the papers on the archive, and I will be happy to discuss about it if you are interested in this topic. So I'm going to move now at actually what is the topic of this talk is I would like to introduce this toolbox that we have been developing in the last couple of years, depiction. So basically this is an open source toolbox. We have an open repo in GitHub that all of you can download. And we have implemented, so of course, not all of the methods for interpretability because there are too many, but at least the ones that seem to be more popular or more useful. So when you look into this repo, well, there are a number of folders which you have also some workout examples. You have some Jupyter notebooks with data and examples that you can really put your hands on as soon as you want. But if you're looking into the models, you should go into this folder, depiction. And here you are going to find a description of the models that have been implemented. And as you can see, we have different types of models. They are categorized in different folders according to the type of model. So you have some of them that is based on backpropagation through the network. Others that are based on a perturbation method, as I described before. For instance, the occlusion methods will be some type of perturbation method. But we have others that are more inspired on traditional machine learning approaches. And also you might want to look into this folder, workshops. So in the last couple of years, we've been running a tutorial on interpretability in several well-attended conferences. And we have gathered a lot of material that you can find here in this folder. So we have slides, hands-on exercises. We have also webinars. So everything is here, also some hands-on data. So I encourage you to play with it if you are interested. Okay, so now I'm going to give you a few examples of what you could do with this toolbox. And yes, I think I have the time, so I will, well, maybe I not so much time, but I would like to introduce at least this example. So we wanted, this is a data set that was published in 2015. It's 3 million single cells profiled with CyTOF, and we have 13 markers from this bone marrow data set. So basically, this is the classification given in the original paper. So you can see that they have profiled different types of cellular populations. So our first task was to develop a deep learning model that would classify the cell type of each cell. That was rather easy because we have many cells, 3 million, and we have 13 markers. So this is the dream problem for a deep learner. So that was the single task. And then what we wanted to do was to apply these interpretable methods to try to extract which markers were important to classify each sample. So this is an example of here we run as a subtype of models. These are called gradient-based. These are based on backpropagation. And this is the marker that was ranked top by each different method. And here in each marker, here in color, you can see the expression level of each marker. This is a logarithmic scale. So bright color means that it's very highly expressed and dark color very lowly expressed. So actually, when I look at this table, I was very happy because actually we do quite well. So just let me start with the simplest example. If you look at mature T cells, as Denis explained in the previous talk, actually T cells, at least effector T cells, come in two flavors. You have CD4 positive or CD8 positive. And actually, this is a binary switch that you have your one receptor or another. And you can see here that at least for CD4, 4 models out of 5 pick this marker CD4 as the top marker to classify the cells, and the same for CD8, 3 models out of 5. And the other marker is also quite important. It has to be highly expressed to predict this category. And here I only showing the top marker. If you go into the second rank and third rank, actually you see the reverse switch. It tells you that to be a CD4 positive, you also do not have to have high expression of CD8. So that was quite nice to see. Another example that also worked quite well is the plasma cell is one of the main feature has to be really high expression of CD38. And here you have with a very bright color, it's really, really highly expressed. Yes.
You have 1 minute, sorry.
Okay. Yes, maybe I was a bit slow. So well, just to tell you that we are doing quite well in another number of classes. And so this is just the first assessment. As you can see, we do quite well. You have, sorry, I'm rushing here, but you have the information in the PDF if you want to look at it. Also, we did the same example with image analysis, and actually we were able to classify cancer and non-cancer and strike which was the features important to classify a cell as cancerous as cancerous and normal cell. Okay, so oh, there was a slide missing, but doesn't matter. So I would like to conclude since I'm running out of time. So the challenge, I hope I have convinced you that this deep learning is achieving breakthrough performances, but these models is very complex and very highly non-transparent, and this creates problems. So I hope they are convinced now that the topic in trying to develop models that are transparent, or at least that we have ways of extracting insight of this model, is highly important. Sorry. So we have done quite a lot of work in trying to develop metrics to assess what is a good explanation. Even though there's no such a thing as universal explanation, at least we can put forward a series of metrics that will tell you how good or bad this explanation can be. And then the last thing I have mentioned the work we have done with depiction, which is an open source toolbox for different interpretable methods. And I have shown you this example of CyTOF. This will I just rushed through it, but I encourage you, if you are interested, to look at the PDF and come back to me if you have questions. And the idea is that through these techniques, we can really extract insights about the mechanisms behind the classification. And with that, I would like really, well, this is our webpage. So this is again the webpage of where we have all the data, the repository. And also I encourage you to look at our webpage because also we have another number of softwares that are open source for which you also have many open web services. And with that, I would like to thank the people, especially the whole team, but especially the people who did the work here, Amphi, who just was very hardly working on this, and also Sabina Kim. That, thank you for your attention.
Thank you very much, Maria, for your talk, and I'm really sorry for the time constraints. So very quickly, a question. What language is depiction built on?
Yes, so it depends on the model. Typically, we have taken the models as, and they were taken for the models have been implemented by the authors. So some models are run on PyTorch, some of them are running on TensorFlow. So we have both versions. Typically, we do not re-implement the models. We just take them and write a wrapper to write them in a Jupyter notebook.
And very quickly, talking about the evaluation, have you found a popular example? Best explanation does not really fit your criteria?
Sorry, the sound was breaking, so I did not understand the question.
Maria, I don't know if you.
Yes.
Okay.
So the sound was breaking, so I maybe stopped sharing the screen. So sometimes I did not understand the question.
Okay. Okay, so talking about the evaluation metrics, have you found a popular example where the best explanation does not really fit to your criteria?
There is not something I can cite right now, but actually there are many examples where we have a lot of cognitive biases in which we think we know what it is, but actually our thinking is biased. I don't know who has the question, but I will be happy to discuss outside this talk.
Okay, so you can use actually the discussion forum to continue chatting. Thank you again.
Thank you very much
for your interesting talk. So now I'm going to ask Dr. Paul Macklin. I don't know if you see my screen.
Great, thank you. Is it okay if I share my screen, Anna?
I'm sharing mine. I would like to introduce you first.
Oh, I'm sorry, my apologies.
It's okay, but do you see my screen?
Yes.
Okay, good. So we are happy to have Paul with us, who is an associate professor at Indiana University in the Department of Intelligent Systems Engineering. So Paul's research focuses on PhysiCell, open source multicellular simulation framework, multicellular and multiscale systems biology, cancer biology, infectious diseases, and tissue dynamics. And today he's going to present the community-driven effort for building a multiscale model for SARS-CoV-2. So now I'm stopping the sharing of my screen, and Paul, you can start sharing yours.
Great. Thank you very much. And hopefully this works. Okay, so hopefully you're seeing my slides. Thank you very much for the invitation to talk and share this work here. I'm honored to present really what is not my work, but work on behalf of a large coalition of multidisciplinary scientists. Mathematical modeling, of course, is kind of at the foundation of this, and computational modeling, but also a whole group of pharmacologists, immunologists, virologists who are working together to build this work. We found actually that we looked at our Slack workspace statistics, and on any day we have about 40 regular contributors from 20 or more institutions working together on this effort, spread across North America and Europe primarily. And I just wanted to point out the two student leads in my group on this, Michael Getz from India, who just joined us as a virtual postdoc here at Indiana University, and Yafei Wang, who is a PhD student who I think will be on the job market in about a year. And they're doing just fantastic work, and I'm grateful to work with such a large coalition. I also want to thank some of our sponsors for this work. This is kind of an interdisciplinary tale where the methods we developed were primarily for our cancer research, funded by the National Cancer Institute and the Breast Cancer Research Foundation. The National Science Foundation helped us fund us to deliver methods to transform complex mathematical models that run at the command line and share them in a Jupyter Notebook interface and share them as cloud-hosted models. And then for this particular work, the Jane Koskinas Ted Giovanis Foundation for Health and Policy helped give us a Jumpstart grant to help jumpstart this coalition. So thank you very much to all of that. So first, to kind of introduce the problem, which I'm sure this is all quite familiar to most of you, but COVID-19 is just an extremely multi-scale problem. At the subcellular level, you have, of course, all the dynamics you'd expect. Viral binding to receptors on the cell surface, and then endocytosis or viral entry. Once a virus gets inside of a cell, it has to be encoded and start replicating and creating viral proteins, and then assemble those into viral particles or virions, and then eventually send those back out of the cell in exocytosis to release the virus. Of course, as part and parcel of that, ACE2 receptor has been identified as one of the main entry points for this virus. And so this couples to the normal processes of ACE2 receptor trafficking. And then once the virus gets into the cell, it doesn't just replicate like a factory. There's a whole lot going on there. So you might imagine from Jonathan Karr's earlier talk here this morning that there's a whole lot of signaling going on in those individual cells, and they respond to the presence of that virus. But the most important things that we worry about is the cell death response. So the cell can only handle so much of a viral load before it just wears out and gives up. But there are also other feedbacks like interferon signalings, where the cells will secrete signals that both inhibit the virus within that single cell, but also kind of give an early warning to the neighboring cells that there's something bad in the neighborhood and they ought to slow down some of the processes. And that has an antiviral effect as well. Then, of course, at the cell level, infected cells, again, they die, but they can warn each other's cells through interferon signaling. Immune cells are wandering around and they can phagocytose dead cells, and immune cells can attack infected cells. Then at the tissue level, you get the spread of the virus through the tissue. This kind of like this growing region of infection is called a plaque. So that infected region spreads. And then following behind that plaque, those cells start dying either by the virus killing off cells or by the immune system coming in and killing off cells. And you get tissue damage, which leads to a whole other cascade of effects. And then immune cells coordinate within this environment primarily through secreted factors, but also through contact. And then at the systems level, at the whole body level, you have immune cells that are getting signals in the lymphatic system and go through things like expansion of the immune system and producing antibodies. And then those then traffic to and from the immune system to the local sites of infection. So there's a whole lot of multi-scale dynamics going here just to understand what's going on in a typical infection, whether it's COVID-19 or any other viral infection. So this is definitely a complex system. And what we want to do is use mathematical modeling and computational modeling to help understand the system and find better treatments. So for example, where are the vulnerabilities in the system? Suppose we can come up with a perfect inhibitor for some process. Would it make a difference? And it's the multi-scale balance of all these processes that really tells us what's going to happen here. So we're trying to build a framework that is rich enough to study these dynamics to say, if I tinker over here in this bit of the complex system, what might be the side effects that percolate through this complex system? So our approach here is team science, that we're recruiting, again, virologists, mathematicians, pharmacologists, and others with unique domain expert. You can't do mathematical modeling of a problem like this without the right domain expertise. And so we want to have these domain experts team up with mathematicians and computational models to build submodels of this in parallel and integrate them into a comprehensive model. If you take the more traditional approach of aim 1, aim 2, aim 3, and develop the different subparts of the model one at a time, we're not going to have anything working for years. So we have to be able to develop in parallel of dividing into sub-teams and coordinating the work. And then trying to get community feedback based upon cloud-hosted versions of the models and, of course, rapid dissemination of preprints. Everything we do is based upon an open source tooling. PhysiCell is open source. I'll show you that in just a moment. Later on, as we get farther on, we can use EMUs, which is out of Argonne National Lab for massive large-scale investigations and high-performance computing. We have developed a toolkit called XML to Jupyter that reads the XML configuration files for a given C++ based model, creates the Jupyter widgets and a Jupyter notebook that acts as a GUI, and we can host that in the cloud on NanoHub. And then, of course, we're trying to take a cue from engineering to use rapid prototyping dissemination that we coordinate very frequently. We develop in parallel. Every now and then, we put the whole thing together into an integrated package and make a release. And on GitHub, using all the typical practices, Zenodo DOIs for software archives. And then we update the preprint and spread it on bioRxiv to make sure that the community is seeing what we're doing, we're being transparent, and then we can take feedback from the community into the next iteration of the model. So we try out a version, put it out there, see what the feedback is, see what works, what doesn't work, and then set priorities for the next iteration. So for a project like this, you really have to think about the life cycle. And so we've really developed this as a series of phases towards a self-sustaining community. So phase 1 of the project was really just us in our lab saying, is this even worth doing? Is this something that we can make a difference in? And so we said, let's do some rapid prototyping and proof of concept modeling and build some infrastructure and start recruiting the team. And so we recruited a great coalition that I'm honored to work with. And as they come in and they come up to speed of how to use the software, how to contribute and self-organize into sub-teams, we did kind of most of the lifting here. We built the software infrastructure. We populate it with placeholder models that kind of get the generic qualitative behaviors that we want to see in the dynamics. And then we move on to the phase we're in now, where the community starts to take it over. That the sub-teams that self-organized in phase 1 are now going to take over those placeholder models that we helped build and start replacing them with domain expert-driven models that really start getting the correct biology and other aspects into this problem. So we'll show some great examples of that as we go through the iterations of this model. And then phase 3, which is the eventual goal, is that after a while, this rapid prototyping had better slow down. We start to converge towards a model that everybody here is happy with, and we put it out there for widespread community use. And there is our hope that multiple independent teams are going to make use of the software and use it in their own independent investigations. And then we're going to act as a data clearinghouse and as a results clearinghouse to say, let's see what worked. And as people get their data in, we'll refine the calibration and model parameter estimates and put those out there and update the model that is released and available to the entire community. And then, of course, bug fixes and feature refinements as you need to based upon what we're hearing from the teams, what did and didn't work. And it's our hope that this is not just a SARS-CoV-2 model, but a generic framework that's going to be available for modeling the next pathogen that comes along. We don't want to start from scratch every time we model a new novel pathogen. So that's kind of the goal here. And so we were quite fortunate to have a rapid coalescence of our community. Somewhere in late March, we did a little bit of overnight work of a really basic viral replication model spreading in a tissue. Within a few days, we were able, actually within 12 hours, we were able to get the first version of that shared in the cloud using these frameworks that we developed in the past. So there's really great value in developing frameworks to facilitate rapid turnaround modeling like this. And within the first week, we were able to kind of start nucleating our coalition and put out the first version of a community-driven preprint of bioarchive. So we're really quite grateful for that. And now that we've grown to so many contributors, it really takes a fascinating coalition management that we really do have to organize into teams. Every week we have a coalition meeting where we get updates from all the major active sub-teams. And they continue developing in parallel, hold their own weekly meetings at their own as their own needs dictate. And then as we get towards the end of an iteration, we start gathering up the software and integrating it, preparing it for release. And so while we're doing the work on the integration software testing, the sub-teams then switch their focus to updating the scientific preprint so that we can disseminate the results and document what we're doing. And so we take this nice iterative approach of going from parallel development to assembling, and everyone's working on the preprint. And it's been pretty nice so far, but of course, learning experience to organize a large distributed team working over multiple continents, and everyone here is basically a volunteer. And so that also changes the time scale of work. We have quite a few active sub-teams. Probably the most active team is in the tissue immune response, modeling individual agents of macrophages, T cells, dendritic cells, neutrophils. And then now systemic immune, so that's kind of the response of an infected tissue. And then kind of the off-screen response is the systemic response of how the immune starts expanding in response to the signals coming from the local model, and then acting as a cell source and sink for immune cells to start being recruited back into the tissue. Each infected cell needs to have its own response model, both death from the viral load, changes in their phenotype based upon things like interferon signaling, and death responses from interactions with the immune system. Of course, you need a viral replication model, and so we have a viral replication team trying to improve the realism of that model. We have another team working on tissue damage and repair, particularly focusing on fibrosis right now. And then we have some more technical teams. So we have an SBML team that's working on improving the support for SBML models in each of the individual cell agents that we'll show you in a moment. And then a pharmacodynamics team that wants to model the transport of drugs and then changes on the parameters in these different components based upon what we know about the chemistry and the pharmacodynamics and pharmacology of those drugs. So let me give you kind of a snapshot of the modeling tools that we're using and then go back to some of the early results we've gotten from this coalition so far. So the first thing we realized a long time ago is that biology is driven by a lot of diffusion. And so unlike many problems where it might be just one or two PDEs, you're talking oxygen, glucose, waste factors, a drug or two, acetylene factor or two, and you're talking very easily 5 or 10 or more diffusion equations in 2D or in 3D. And so we spent a little bit of time trying to make a more scalable vectorized diffusion solver that solves a whole vector of diffusion equations at the same time. Worked really hard to make sure it scales nicely and that it's still accurate, and that it ultimately, you can simulate a few cubic millimeter of tissue even on a desktop workstation with 5 to 10 diffusion equations or on a single compute node. And this is cross-platform compatible. We run it on Windows, Linux, OS X, compute nodes, and it works quite nicely, and this is in bioinformatics. Then on top of that, we built an agent-based model system called PhysicCell that takes that BioFEM, which had cell diffusion and communication with static point-based sources and sinks, and turn those point sources and sinks into cell agents, each of which has an off-lattice position, mechanics-based movement, individual cell processes for the classical cell behaviors like division, cycling, volumetric growth, death, motility, and then signaling, controlling the sources and sinks in those diffusion equations. The cool thing is you can have individual custom data values and custom functions applied on a cell-by-cell basis, and you can even change them dynamically at runtime, so it becomes quite flexible. This is also cross-platform compatible. We have undergraduates in our lab running it on their laptops on a Mac. We have others working on Windows or Linux, and then when it comes time, we send it out to HPC if we want to do really big investigations. So as an example, here's a model of immune cells. These are the red ones going in and attacking a tumor cell with a heterogeneous immunogenicity. So the yellower the cell here, the more immunogenic it is, the more attractive it is to immune cell. These are kind of like T cells attacking a tumor. And those cells are also faster in proliferation. And so we just show an example for cross-section of tens of thousands to hundreds of thousands of cells interacting in a 3D environment, and you have very complex dynamics that emerge. And so this is just one example of taking this framework with some very simple model rules. You start getting something that looks like a CAR T-cell therapy or immunosurveillance. And you can actually, I wanted to point out that this model is included in every download of PhysicCell, so you can try it yourself. And there's an online version of it at this little QR code over here. And I believe they're going to share a copy of the slides, so you can click on the hyperlinks later to try this out. Well, one model is a demo. To do science, you need to really do more replicates and explore your parameter and design spaces. And so we looked at this model and said, well, there are 3 parameters that we care about, at least looked interesting to us. And so suppose you have low, medium, high values of those parameters. So it's 27 parameter sets. You better do a few replicates per set. So now you're talking about 270 model runs. It takes a couple of days per run on your desktop, though that's a year and a half of computing. So clearly you can't do that kind of a study lightly unless you have high throughput computing. Fortunately, we talked to Argonne National Lab and just ran the entire study all at once over a weekend on a Cray and got some really nice results out of it. So this shows an example that if you work on making a computational method that's easy enough to develop on a desktop, but fast enough to run on one compute node with OpenMP parallelization, then you can use your high throughput computing infrastructure to start doing really interesting studies that go across parameter spaces. And just one more quick point here is that you can ramp it up to even higher dimensional design spaces. So we looked at, say, add 3 more parameters in a 6-dimensional design space and look at a series of design problems. Can you control the cancer with your immune parameters? Can you get it actually to go into remission by getting smaller? Can you optimize the treatment? Well, even on HPC, you can't brute force a 6-dimensional design space and try every single possible combination of parameters. So you need to be guided in your exploration. And so we used active learning. And so there you say, for any design scenario, turn it into a binary decision tree classifier. Did I meet my design goal or did I not meet my design goal? And then you run batches of simulations and choose them with your active learning to help resolve that decision boundary. And so what this allowed us to do is to cut down the number of simulations per design scenario by a factor of 10 to the 3, which means that rather than spending all of your compute resources just to find 1 or 2 design scenarios, you can actually do a nested set of design scenarios, starting getting at the topology of your design space. And the bonus there, of course, and this really hooks very nicely to Maria's talk just a moment ago, is that if you use something like a decision tree, your results are much more interpretable because you can use the Gini coefficients to rank your parameters and see what's important in a particular model. So there's just kind of examples of the tools going just first from simulating diffusion, adding agents on top of that with very customizable rules, and then ramping up the explorations to HPC to really explore a high-dimensional complex design system. And that's going to have great implications later for things like optimization, particle swarm optimization, or even calibration and things like this. So now let's take this toolbox and start focusing it on COVID-19. So we went through a series of design steps. We have a first prototype that we developed, again, the one I showed you just a little bit ago, overnight, and then spent about a week trying to document it. And so for this first proof of concept model, we start with just a single monolayer of epithelial cells. So imagine that you have, say, a layer of lung tissue resting on top of an epithelium. Imagine that if you're facing outward from the screen, you're in the lumen. And if you're going down into the screen, then you're on the basal lamina and then getting underneath into the endothelium. And so that's kind of your mental image of what you have here. And as a first proof of concept, we said, what happens if a single virus particle lands in the center of this tissue and infects the cell? And so to combine the different model elements, we start with a PDE-based model of the environment to say, let's treat virus particles as a diffusing field. And so you have your classical diffusion over here, and then you have no decay here in this case, but you have at each individual cell, you have individual cell uptake. I think I'm missing an element here. There should be one more part, but that individual cells uptake virus, like a classical reaction term, and later on, infected cells are going to be exporting virus back into it. So you have cells acting both as sources and sinks of your virus particles. Okay, so now you have cells that have virus particles diffusing through your tissue. They land on a cell and they get removed from the environment and then pop up here in a compartment in a submodel for the individual lung epithelial agents. And so loss from the diffusing field is a gain to the internal compartments. So V is internalized viral particles. And then at some point, those virus particles get released from little vesicles and they get encoded. And so now you have uncoded virus particles. Those uncoded virus particles get prepped into usable RNA, just in a really simplistic model here. RNA eventually creates protein. So the more RNA you have, the more protein you make. Now this is neglecting complex things like amplification, but it's a start. That protein may have a lifetime, but now proteins are assembled into assembled virus particles. So that's the A compartment. And then eventually those are exported back out to the PDE, back into the source term over here. And so then you have to use mass matching terms to make sure that you can get a mass conserving uptake rate and export rate. So you can match what's going on to the PDEs to what's going on in individual cells. So each one of these individual cells now has a copy of this compartmental model of viral replication. Now that just gets virus being created and being released into the environment, but you also need the cell response. So it's a very, very, again, it's a placeholder model, and this is coding we did overnight. We tie the cell death rate to the level of assembled virus particle in that cell, and use kind of a hill type function that the more virus particle you have in there, the greater the death rate, which means that starts reducing the mean lifetime of that cell. And so on the right, we show an example of this. Say, so you can see how one virus particle lands in the middle. The more yellow this simulation is, the more infected that cell is, the more assembled virus particles that cell has. So the shade from blue to yellow is the viral load. And you can see how that one virus particle lands, then the infected cell region spreads out. The next thing you see is any one of these black cells is a dead cell. So that's a cell that has responded to the high viral load by a death response. And then as that dead cell shrinks, you see a white region left over. So that's the damaged tissue. So you can see there that you have the virus particle landing, and then that plaque is spreading out, this infected region. And then following that, the tissue damage spreads out kind of concentrically, as you might expect. So a basic placeholder proof of concept model developed in a matter of hours was able to start getting some of the classical features of the spreading viral plaque. So that was version 1. And of course, the point of a model is to start exploring to say what happens if you change things. What are the impacts of different model parameters? And so we gave a look and said, for example, if you slow down viral diffusion, as you expect, the plaques spread more slowly because the virus particles are spreading more slowly. So fewer cells are getting infected right away. And the other thing you might expect, which we definitely see in the model, is that the plaque boundaries are sharper. So that, of course, makes sense. We had a look to say what happens based upon whether apoptotic cells or dead cells release all their virus particles at lysis, because some viruses do that and some of them don't. And at the beginning, before we had recruited all of our domain expertise, we were worried about that parameter. And we actually found that at the end of the day, it had very little impact on the overall multi-scale dynamics. Well, it maybe impacted quite a bit on, say, the local dynamics. It really had almost no bearing on how quickly that plaque spreads through the tissue. On the other hand, if we made the infected cells a little bit less tolerant of a viral load, that means they die earlier, which means that they have less time to create more virus particles and they don't pump them out. And that had somewhat of effect of spreading the speed of that viral plaque. Likewise, you can look at the mean survival time of an infected cell. There, it can tolerate a higher load, but they may apoptose a little faster. And there we found that the tissue damage was much faster, but it didn't actually slow down the spread of the plaque, so which was kind of surprising to us. And that's just where these nonlinear emergent effects start popping out. So that was the first version, proof of concept model. By version 2, the second prototype, there we had started recruiting in the domain expertise and started refining the model assumptions. But here we're still largely building infrastructure and letting the teams coalesce and form sub-teams. And so this one we focus more on software infrastructure, making the model format a little bit more modular. But we did add one thing. We added ACE2 receptor dynamics. And so here now, we added another compartmental model, an ODE model, to each individual cell agent that has basically 4 compartments. You have unbound receptor on the external surface. You have bound receptor, which means a receptor that's bound to a virus particle. Then you have internalized bound receptor, so it's been pulled in. And then you have unbound internal receptor. So we kind of go through a cascade that you have receptors on the cell surface that are reacting with the diffusing virus field. When they react, that forms a bound pair. That bound pair gets internalized to move into the internal compartment, then it gets released. And so at the point that internalized receptor becomes unbound, that becomes a source then to the earlier viral replication model you saw in the version 1 model. Then that model continues from there. And then eventually we have unbound internalized ACE2 receptor gets returned to the cell surface in a trafficking model. So now you have that ACE2 receptor trafficking model that is tied to the internal viral, to the uptake of virus particles, and then the viral replication model. Otherwise, everything else is the same. The other thing we changed is, we talked to the virologist, said, well, you never just put one virus particle in the middle of a tissue. They land randomly there. So we knew that we had to be able to model a variation in, say, the multiplicity of infection, which is the ratio of virus particles to target cells. And so here we have virus particles landing randomly in the tissue and then forming a variety of plaques that start growing and then merging, and then the tissue death follows. So far we have no interferon response in this model, and we have no immune response. So this is like an extremely immunocompromised patient, and every infection is is not good here. There's no way to contain this infection. And of course, we can look at other parameter values, but let's, I think, continue forward to the next version of the model. So the third prototype we released very, very recently, well, was very recently, but towards the tail end of July. And so this one had some major changes. The tissue immune team had really ramped up and started adding their assumptions in and said, well, we really need to start modeling that local immune response. So we started off with the same lung epithelium model that we talked about before. No changes in that model in this iteration, but we added a few new types. Macrophages, let's see here, I'll kind of scroll this back. Macrophages are these pale green cells that you see here. Oh, nuts, I think copy paste started killing some of my colors here. So we have some resident macrophages that wander through the tissue. And when they find a dead cell, they activate and start secreting a pro-inflammatory factor. Also, activated macrophages will continue to phagocytose dead debris as it finds it. Now, as that pro-inflammatory marker accumulates in the tissue, so as more, which is going to scale with the number of activated macrophages. So here's an activated macrophage, these pink cells here. Those then start recruiting in neutrophils. And so these are the cyan cells that you see appearing, and T cells. And so let's scroll through here just a little bit. We'll look for the dynamics. Here we see some neutrophils. And in this model, neutrophils can phagocytose dead material, and they can ingest or absorb viral particles, which kind of helps a little bit to slow the spread, although that wasn't terribly significant in our model. And then later on, you're going to see some red T cells recruited. Let's see here, a little bit farther on. Yep, so here they are. Red T cells come in, and they are looking for infected but not yet dead cells, and they will form an adhesion. And the longer a T cell's in contact with an infected cell, the greater the probability of apoptosis, which gives them kind of like a mean contact time for apoptosis. The cool thing here is that as an emergent phenomenon of this pairwise type of interaction, there's nothing to prevent multiple T cells from adhering to one infected cell. And the more T cells you have infected, the faster this response is, which is very similar to what we see in CAR T cell therapies in cancer, that you can see multiple T cells attacking a single cancer cell. And if you have multiple T cells attacking it, the thing dies a little faster. And so we actually have as an emergent phenomenon from these very basic model rules here. Everything else is the same, but we have chemokines now secreted by infected cells to help attract T cells. And then we have the cytokines being secreted by the macrophages. So now, with such a model, we can start looking at different T cell strategies. So, for example, what happens if you recruit T cells faster? And so here, we reduce the threshold of when T cells get recruited. And you can see that, they're definitely gobbling up the infected cells a little faster, but we do have some model shortcomings in it, so that doesn't quite bring the infection under control, although it is very, very slowed down. Likewise, we could say, well, what happens if they're recruited at the same rate, but now the T cells are just much more effective at killing an infected cell? And you see very, very, similar responses that they're able to slow down the spread of these plaques here in the tissue. The other thing we said is, what happens if, for some reason, there are already T cells there to begin with? So maybe this is a tissue that is adjacent to an infected tissue. So T cells have already been recruited. Or maybe you were infected and recovered earlier, so you have some T cells resident in the tissue that remember what they've seen. It's a really simplistic model. And here, you can see that they're really aggressive at attacking those cells earlier on. And then you have the nonlinear feedbacks of the system, that the T cells are killing things faster, which means macrophages are finding dead material faster, which means that macrophages are activating sooner, which means that macrophages can recruit more cells sooner. So you see a ton more recruitment of immune cells just by having some resident T cells. Now, whether that's right or wrong, we don't know. We need to start working with more domain experts on this. But this shows you how the nonlinear interactions from very simple model rules can lead to some very interesting emergent dynamics. And there are other results we looked at. We can put them side by side to really look at the differences. And of course, the beauty of a mathematical model is you can post-process it and look for differences in the curves of, for example, how the titer changes over time, the number of virus particles in the tissue. And so that's something we've looked at quite a bit in version 3. So we got a lot out of this. And we also saw, based upon our coalition and community feedback, there were a lot of things missing, that we definitely need those feedbacks, say from the interferon signaling, if we're going to do a more realistic modeling of the tissue. Right now, basically, it's still fairly dismal fate for this infected tissue, that no matter what, even with this immune system here, it really slows down the infection, but doesn't completely hold it off. So we can say, well, what's missing? Well, we definitely need the interferon signaling. We're missing a few different immune cell types. We're missing the off-site immune expansion. So let's think about those. The other thing is what we found is that the classical technique of modeling viral replication with a system of ODEs just doesn't work quite right in this kind of a context. Modeling it as an ODE works great as a mean field for a bunch of cells, but on the flip side, taking that mean field model and putting a copy in each individual cell doesn't quite discretize it right. And so what happens is that you have these continuous source terms that are uptaking virus particles from the domain, and now you have, say, 1/10 of a virus particle in a cell that all of a sudden can make RNA and protein and create and start viral synthesis and export. And so that's not realistic. You can't have 1/4 of a virus particle making virus particles in an infected cell. You can't be 1/4 infected. And so we need a better discrete continuum analog here to say that really it's a probabilistic sampling, that if a cell is sitting in a viral field, it has some probability of recruiting 1 or 2 or 3 virus particles. And only when you have enough to do an entire virus particle recruitment, then you pop it into the compartmental model and go as you would have before. So that's really prompting some changes in version 4. So I'll get back to this in a moment here. But one thing that's very nice is that we can learn from other fields, and we're taking expertise from all sorts of fields and computational domains and applying it to a new problem. And then when we're done here, all these fields are going to learn from what we have here. We're going to have a whole lot of immune models to deliver back to our kind of our primary problem in cancer, because we're going to have all these immune cell models already developed. So the 4th prototype is ongoing, improving the tissue immune response, improving interferons, and having an off-screen model. Thanks. I think I'm on track, actually. And we're going to start adding these components. So as an example here, some great work led by Adrienne Jenner at the University of Montreal. She's now working with her team of immunologists and pharmacologists and virologists to improve their model of the tissue immune response. And so we're saying, well, we know we're missing macrophages and some of the polarizations of them. We know that they interact with T cells to change their interactions. We know that there should be dendritic cells that are sampling infected cells and reporting back to the off-screen, systemic immune model, and they should be driving, the changes in the behavior. So they've already started prototyping in their individual version of the model all these components here. So now we have a variety of macrophage polarizations and T cell behaviors and neutrophils, and then these active and inactive dendritic cells. The next step is going to be connecting this with the other components of the model. That version 4 is going to have a brand new model of the lymphatic expansion of T cells and other immune cells. And that will become the source and sink term for immune cells landing or leaving from the immune tissue, from the local infected tissue. So I'm really grateful to have this opportunity to present this coalition's work. We have a long ways to go, but we are quite thrilled to have gone from 0 in March to this, and we hope this becomes a valuable resource to the community. So thanks very much for your time, and please do have a look at the preprint, and of course, we'd love help. So, cool.
So thank you very much, dear Paul, for your wonderful talk and for keeping the time. So we have many questions already. Have you found hypotheses that immunologists have said we just do not know, or better, that they have opened new research avenues?
To an extent, right? For example, we have, one thing I'm very happy with is that this model is already being used by independent teams in grant proposals and in spin-off papers. So it's nice to see this starting to nucleate independent work in teams. One team we worked with was doing some calibration to some antiviral work, and that really led us to change quite a bit some of our assumptions of how the virus spreads, and also some of the negative feedbacks of it. So one thing we found in particular is that an antiviral might knock down the titers quite a bit, but because of the nonlinear dynamics here, we found that there's no way it could possibly be a perfect inhibition to have any measurable titer at all. You still have to have replication exocytosis to match those numbers, and it was actually quite interesting to get that matched. We're also really seeing, I think, the critical importance of the interferon modeling here, because we really just cannot match the models without it, and the observations of the data.
So how do you assess the validity of your simulations? How do you know that they are biologically relevant? Do you have data to compare them with?
So that's a really great question. And I would view this kind of in 2 levels of validation. The first level is we have a community of domain experts. Effectively, with 40+ people here across domains, we're giving the same peer review every day. And the first level here is that we have domain experts in immunology and viral biology who have been learning from their own observations for decades of what a virus should and shouldn't do. So our first step is, can we come to a point where this model, at least qualitatively, can reproduce the right behaviors that are observed by people in the field at the right time scales? It's kind of the physicist's point of view, right? That you have to get the right behaviors at the right time scales, at the right spatial scales. And that's kind of like reaching the starting line of a race. Say that once we have a model that can reproduce at least the right qualitative behavior, now it's time to start working on the quantitative validation. Now, if we wait to release this package until quantitative validation, the pandemic will be over. We'll probably have one more pandemic over before we get to that stage. So what we really want to do is get to a point to say, we think this is a model that's worth working with, that the team of experts say that this is giving reasonable behavior. Let's release it to the wild, and let people do their individual investigations, and then bring that data back in to start validating components of the model. And I think that's the only way we can do this and still get progress fast enough to make a difference. But over time, we expect to calibrate and quantitate and validate individual parts of the model and eventually the emergent dynamics.
So is your host tissue cell agnostic? How do you model it? Because for
So that's a great question. You know, what makes that a lung epithelial cell and not something else, for example? What makes that SARS-CoV-2 versus just flu?
exactly.
And right now, I'd say it's fairly generic. As we calibrate the model parameters better and better, I think it's going to get a little bit more virus specific. But really, there's quite a lot of shared biology between these different viruses and tissues. I think where it starts getting more specific is when you start modeling, say, a specific receptor, a specific death response, specific survival times. The other thing that's going to get interesting is really because we know that SARS-CoV-2 can infect endothelial cells. We should probably introduce a copy of that in the underlying endothelium and then tie that to the edema. And that's when it's starting, I think, start getting a little bit more specific to this particular virus, this particular tissue. We could also try other tissues like myocardial tissue, for example, where you have other receptors. And they have different lifetimes and survivability to a viral load. And there are a whole lot of hypotheses you can start testing, and I think start making it more specific. It's a really good question.
And how difficult it is to put together all these sub-models, and also from the management point of view to actually coordinate all these different labs across the globe, actually, and make something that actually works at the end.
That's got to be one of the biggest learning experiences I've had in my entire life. Usually, I don't think I've worked with a team bigger than 4 or 5 or 6 people before. So 40 to 60 people on a day-to-week basis has been phenomenal. And I'm grateful to work with such great people. It has been hard, right? Especially because we didn't design our model framework to scale to that size of a team originally. And so a lot of it's still kind of manual, where we have the different teams that develop their models and kind of imagine forking it, and then we pull it in together. And there's a lot of manual checks right now. We could use some techniques and help in automating that process. But for now, we're doing a very manual curation, which means that the model integration stage of any version of the model takes a lot of time. But we're trying to be very, very careful about that. The team management has actually been really quite exciting, because we do have these weekly coordination meetings, but we have such a great group of people that they've come to a point where really they're self-managing all their individual teams. They've decided on their own when to meet, how often they form their Zoom meetings. We have a Slack workspace that helps facilitate it so that everyone can kind of exchange, and we keep it as a very open community. So far, that model has worked quite nicely, and you start seeing who the emergent leaders are, and you start working with them. We try to have each sub-team designate a scientific lead and a technical code lead, so that when I have a question on integration, I know who to talk to. So for example, on tissue immune, I talk to Adrienne Jenner. When I talk to bioreplication, I talk to my PhD student. And so we know who the points of contact are to kind of smooth the interaction between the teams. But it's been an experience. I'd like to see it replicated. I think we could do this for other diseases, and I think we should. And it's neat to see what can happen.
So, and the last question, what would differentiate your model for SARS-CoV-2 from SARS-CoV-1, for example? Is there something that makes it specific to this particular virus?
Right, and that's quite similar to the earlier question. Right now, I'd say we're a little bit more generic on general viral dynamics and immune dynamics. As we start calibrating the timescales and the parameters and the individual cell behaviors, based upon the observations, then I think it's going to get more specific. I think that the thing that makes it quite specific is when you start modeling ACE2 receptor dynamics. In particular, some of the interesting dynamics that are emerging that, because you're, again, like endothelial cells can get infected, which means you're going to have a lot more risk of edema in this model than you might in some other virus that can only affect lung parenchyma. And so those kinds of things, I think, will start making an emergent effect and then start making it more specific to the virus. But we have a ways to go there. Again, I think we're reaching that starting line where we have something that's, I think, capable of describing those dynamics. And then we can start fine-tuning it more specifically to SARS-CoV-2.
Thank you very much, dear Paul, for your presentation and for your presence here. We need to move on to the third session. So I would like to invite Miguel to start sharing the screen and present the next speaker.
Thank you.
Okay, so very quickly, let me introduce Dr. Joerg Stelling from the ATH in Zurich. And he's a full professor at the ATH Zurich Department of Biosystem Science and Engineering. Dr. Stelling is going to present data integration via metabolic network model. So, Georg, I give you the screen and you can do your presentation. Thank you very much.
Thanks a lot for the invitation, and also Marta for the great setting of the stage. Okay, so it's about data integration via metabolic network models. I want to also point out a little bit what the difficulties with those are. But first, a motivating example. So what we looked at in collaboration with people from medical schools was analyzing gut microbiota. So these are IBD patients, inflammatory bowel disease. And the question was, how can you discriminate between one type of the disease, UC, and Crohn's disease? And usually what is being done, you have the patient here make a biopsy sequence, take the reads, and map to the taxonomy, and then you just look at the metabolic microbial abundances. So what we extended there is essentially take genome scale metabolic models that Marta introduced, and instead just counting how many reactions there are of each particular type, and see whether essentially the potential for metabolism is rather predictive for the biome or the taxonomy. So let me show you briefly the results. And so what you see here on top, these are two cohorts, one and two, which either classify the patients based on these taxonomies, operational taxonomic units. You see the principal components here in this plot explain very little of the variance, so just less than 5%. If you do this at the level of the reactions, the principal components explain roughly 60% of the variability within the population here. You can also see this from the coefficients, so the significance of the discrimination between EUC and CD. This is not significant for when you just look at taxonomy, but when you look at the reaction capabilities, it becomes highly significant. That was the example for cohort 1, and essentially cohort 2 looks pretty much the same. And so a lot more explanation of variability by looking at the reaction capabilities, metabolic capabilities, and not on what particular organisms are now in this microbiome. You can then ask questions similar to those that Marta asked. Okay, so in the two different types of diseases, which metabolic subsystems are overrepresented or underrepresented? So for instance, in Crohn's disease, we find higher lipid biogenesis and biotin metabolism, etc. So these are hypotheses that can now be followed up on more mechanistically, and essentially the same holds for cohort 2. And so the bottom line here is that although we didn't use any advanced modeling, we just took the models and essentially counted reaction abundances, and this can also already help in interpreting experimental data that is otherwise very hard to interpret. So this was the motivating example. To make this a little bit more formal, so I want to explain what we mean by these metabolic models. So these are the genome scale metabolic models. What their content is essentially a set of reactions, a set of metabolites. They're represented in the stoichiometric matrix N here. And the assumption is that the entire system operates at steady state. Then other contents of the model are biomass reactions representing growth and exchanges with the environment. So there are no metabolite concentrations here in these models. We're only looking at fluxes through this network. And as Marta already introduced, so what we get is this solution space here on the right. Given all these constraints, what we typically have is a multitude of possible solutions. So we try to focus this down onto one flux distribution, either by constraining the bounds using, for example, experimental data, or by assuming an objective like this one here that could perhaps mean maximum growth. So the difficulty with this here is when we look at these two ways of integrating data, either flux bounds or objectives, that this is highly non-trivial. And just to give you an idea for why this is the case, this is a toy network here. So we just have an input, this flux here, two enzymes, E1 and E2, and they catalyze reactions in the branching pathway here, so to B and C. So the simulations on the right, they show you essentially what happens when you halve the concentration of enzyme E1 between white and gray. You see the dynamic reactions here in terms of the fluxes. And if both enzymes are approximately at the same level, then you see an effect of this perturbation. So in this case, R2 goes up and R1 goes down, as you would expect. But if you're in a different condition, for example, there is no enzyme E2, then the new steady state will be exactly the same as the old one, as shown here on the right. So it's highly non-trivial to integrate this data, essentially because if you have genomics data, the fluxes are not necessarily proportional to the enzyme levels. And if you have metabolomics data, then the metabolite concentrations are simply not represented in the models. So to try to deal with this, with these problems, we focus on one particular set of problems, namely the question, how can we integrate genomic alterations into these metabolic models? The specific case study here was on tuberculosis. So we're interested in trying to see what the metabolic capabilities that dictate whether a particular strain is infective or drug resistant are. So there's strain-specific variation across the world in tuberculosis, and this could induce metabolic phenotypes, distinct metabolic phenotypes. But now the key question is, how do we identify what the causal genomic alterations, so specifically SNPs, are for these phenotypes? So the setup was as follows. We had a collection of strains that essentially represent all major branches of this organism. They were sequenced, so we know the SNPs here. We did cultivations, or our collaborators did cultivations, so we got the metabolomes, and our main task was then to use constraint-based modeling in order to find out which of the metabolic SNPs are actually functional, meaning that they alter the metabolic phenotype. So just to show you a little bit of the data. So on the x-axis in both plots, you see the different strains. So on the left-hand side, the genetic diversity in terms of non-synonymous SNPs. There are actually, we expected them to be low, but it's essentially comparable to other bacteria. So on the order of 500 SNPs, metabolic SNPs in total in these across all these organisms here, all these strains. And on the right-hand side, you see that this is also reflected in different doubling times, meaning growth rates. Yeah, so we have on the one hand, on the left hand, we have the genotype, on the right hand, we have the phenotype. So the most intuitive way for analyzing this data would be to just do genome-wide association studies. The problem here is that we have only 18 strains and a lot of variants. SNPs. You do the GWAS analysis, in this case, not for the phenotype growth, but for antibiotic sensitivity here. Just doing classical GWAS, then none of these 560 metabolic SNPs has an association with a sensitivity that is actually significant. And the reason is, again, the sample size here is extremely small, so it's 18, and this is pretty representative of many of the problems we have in this field. So what we tried to do then is the following. We looked at the metabolomics data, specifically here for one example. That's how the time course data looks like. So we have, for instance, here pyruvate on the top as a function of time, data points for several replicates, and you see that pyruvate is essentially taken up, indicated by the disappearance from the external medium. Citrate, the same for this particular strain. In other metabolites, such as proline or here oxyglutarate are being excreted. And so we can estimate the uptake and secretion rates of these measured metabolites from the extracellular medium. And what we find there is essentially that there are lineage-specific differences in metabolic behaviors. So if you take metabolites, if you take strains from a different lineage and just look at the difference in the metabolic profile, they're more distant to each other than strains from the same lineage. So this gave a first indication that essentially the genotype and the metabolic behavior in terms of these uptake and secretion rates could be linked in some way. So the proposal that we then came up with is the following. So we want to integrate this into genome scale metabolic models. So we first have to map the SNPs in metabolic genes to the corresponding protein and assume that each SNP can have an effect on the flux of the catalyzed reactions. We don't know the strength of this effect. And what you get then when you make a perturbation, then you have the network response to this SNP effect. In order to analyze this, to predict this, we used a concept we developed earlier. This is called structure sensitivity analysis. So essentially it assesses to which extent each individual reaction in the network is perturbed when you introduce a perturbation at one point, say, through a SNP. So this was the basic idea. Technically, what we do is try to integrate this data by using several different objectives. So we have a flux distribution. I want to predict the flux distribution of strain I. What you see here is the space of potential fluxes. And first of all, the data is not necessarily always consistent between the models and the measurements. So we first want to minimize unexplained flux differences between strains. We also look for a parsimonious solution, namely there's a reference distribution that should be minimal, so as simple as possible. And then we minimize the flux effect of each SNP. So this is step 3. And the assumption here is that we want to detect only those SNPs and their effects that are necessary to explain the phenotype. And that's also how we classify them. If these SNP effects exceed a minimum requirement, then we classify them as functional SNPs, meaning that they are required to explain the data. So the way how this is done technically, this integration of genomic and metabolomic data is essentially that we try to describe the flux distribution of each strain. We have a reference strain. We have the unexplained flux differences just to connect to the data, and the SNP effects. This is E. So this is essentially formulated into one large optimization problem. So for instance, here you have mass balances for each individual strains. They are coupled by whatever we know about the differences in uptake and secretion rates. And then we also have bounds, so the constraints, both on the fluxes individually for each strain, ratios between strains, and then finally the SNP effects. So the computational problem is on the order of 45,000 variables, 28,000 constraints, but it's a simple problem, essentially. So let me show you what the outcome of this analysis was. And so first we asked how many SNPs are functional, meaning necessary to explain the strain differences. Many of the SNPs were predicted to be non-functional, but on the order of 10 to 20% per lineage were predicted to be functional. In more detail, we looked, for example, at this particular SNP here. So there's on the x-axis here, you see the relative SNP effect. On the y-axis are different metabolic subsystems, and the color indicates how many strains have a particular mutation. And so from low to up to 9 different states. So what we found, for example, in glycolysis, there was one particular SNP in pyruvate kinase. It didn't have a huge effect, but it was clearly significant, and it appeared in many strains. So what this enzyme does, it converts PEP to pyruvate and then further to metabolites. And what was essentially known from animal models for MTB is that this pyruvate kinase is essential when the organism grows on media that do not provide pyruvate. So the prediction would be here that this SNP here would have an effect, clearly, but that it would not abolish the ability to grow on non-pyruvate substrates. And that's also what we found here experimentally. So blue bars refer to pyruvate medium, no pyruvate in red. You see a diminished growth rate here in all cases, but all of these strains grow irrespective of whether they have the SNP here. So those are the strains with the SNP or not. So this gave some new insight into now human relevant SNPs for MTB. In the second example, what we found were SNPs that were related to folate metabolism. So this, especially here around tetrahydrofolate, there's an antibiotic called PAS that inhibits part of the folate metabolism, but we also know that some strains are resistant to this drug. So what we predicted here was that mutations in related pathways that feed into THF, these particular mutations here, should make the organisms more PAS sensitive, simply because they block the alternative pathways to THF. And this is also what you observe here. So all these mutants here on the left have a SNP in one of the three enzymes. You see the maximum effect of PAS is essentially much higher than in the wild type with respect to these mutants. Okay, just a brief outlook on how we can try to combine this type of approaches with the sequential minimization to integrate data with the different fields. So we're also interested in metabolic pathway analysis. So the aim there is to enumerate essentially all possibilities of a metabolic network. It's a computationally very hard problem. There exist several different concepts. So what we looked at and proposed very recently was something minimal metabolic pathways. So their characteristic is that they take the constraints from data, for example, into account, and they are also loopless, meaning that they don't generate thermodynamically infeasible cycles. Just to give you an idea of what can be done with this here is we try to use this similar concept of minimization, but to now couple it with pathway analysis to predict essential metabolite exchanges in the human gut microbiome. The nice thing about this approach is it's scalable. So we made a model of human plus 6 microbes, so on the order of 30,000 reactions, 3,000 possible exchanges. And what you see on the right are now the predicted essential exchanges here. So for instance, you see everything in red means uptake and blue means secretion. So for instance, this is a hypothesis on a particular metabolic link where a particular bacterium could provide only to human and to other organisms in the microbiome. Okay, just to summarize briefly, the motivating example was essentially to show you that you do not need complicated computational methods in order to try to make sense of data. Incorporating constraints from genomic metabolomic data is still a bit tricky. So one proposal here is to use a combined model over, for instance, strains of conditions. And the final idea was to combine this type of approaches with metabolic pathway analysis. So the people who did the work were Uwe Oyers and Charlotte Ramon. The first part was in collaboration with the IBD cohort study in Switzerland, and another big collaboration on tuberculosis. And this is the display. Thank you.
Okay, virtual applause. Joerg, thank you very much. Wonderful talk. We have a very quick question, but it's a complicated one. What is the difference between elementary flux modes and minimal pathway? So there are many definitions of this.
There are two differences. So elementary flux modes operate on the unbounded flux space, so no constraints. So it's not a cut polytope. whereas minimal pathways do. And the other difference is that you can formulate minimal pathways in terms of only a subset of the network. So in this particular example I showed you, we were just interested in the exchanges between microbes and humans. So we were not interested in internal distributions. So you get exactly these patterns of exchanges.
So this is related to elementary conversion that was previously defined, I think, by Wagner.
Yeah, so there are elementary flux vectors. So they're related to each other. What we did was essentially trying to simplify this further.
Okay, that's Calet and Schuster. I was okay. There are many. Okay, thank you very much. Okay, we need to wrap up. So I invite the organizer. Thank you, Joerg. Thank you all the speakers. Thank you all the participants and our fellow colleagues. I don't know, Arnau or Anna want to say something else. It was a wonderful workshop.
Thank you for all the presenters that were willing to adopt this new virtual workshop that we didn't quite know how well or bad it was going to be, but I think it was pretty good. I mean, I think we got we got an amazing roster of presenters, and I think they went well. Anna?
So I would like to thank Arnau and Miguel for all their hard work many months now, all the speakers who accepted to come and present their exciting work, and all the people who registered and stayed up to now to follow the session. Even if the setting is a little bit weird, not so interactive as we would like to, but still I think we managed to survive until the end. So I would like to thank all of you and well, enjoy your weekend. And if you would like to say something, mostly the presenters, please open your camera and your mics and do so. We have 4 minutes. So it's not only the participants that are shy and they do not want to say anything, but even the presenters that are reluctant to say at least good night.
Just thank you for organizing the meeting, and thank you for inviting, well, at least me. I think everything went very smooth and very well. Congratulations.
I think we all agree.
Yes. A virtual one.
Good. I think we're going to be kicked out in 2 minutes from this room, so just be aware that we might just go screen to black, you know.
Okay. So I think that I will leave the session before we're kicked out, because you are not willing to talk, and I feel very weird right now. Thank you very much, and see you around. Yes. And for a big shot.
All the link for the attendees of the links is still in the discussion forum if you want to copy paste. Else, just contact any of the organizers and we will send it to you. So yes, good to see you around, and bye bye.
Bye bye.